Welcome to G Fun Facts Online. Today we are going to dismantle one of the most persistent and depressing dogmas of the 20th century. For decades, the central rule of neuroscience was absolute and bleak. The adult human brain was considered a static organ. The belief was that you were born with a finite number of neurons, and as you aged, or if you suffered an injury, those neurons would diminish, never to be replaced. It was a biological one-way street where the best you could hope for was maintenance, not renewal. But that dogma is dead. It was shattered in the late 1990s with the definitive discovery of adult neurogenesis in humans. We now know, with certainty, that the adult mammalian brain retains the capacity to generate functional new neurons throughout life. But the story doesn't end there. The profound realization of the last 2 decades is not just that the brain renews itself, but that this renewal is dictated by the entire body. Your brain is not an isolated command center locked behind the fortress of the skull. It is a dynamic participant in a systemic dialogue. Signals from your muscles, your bones, your gut bacteria, your fat stores, and your immune system are constantly bombarding your brain, instructing neural stem cells to grow or to die. Today, we are doing a deep dive into the systemic regulation of neurogenesis. We are going to look at how the whole body shapes the mind. To understand this, we first need to look at the anatomy of renewal. We call these specific areas neurogenic niches. There are 2 primary ones. First, the subgranular zone, or SGZ, located within the dentate gyrus of the hippocampus. This area is critical for learning, memory, and mood regulation. Inside this zone, we have radial glia-like cells acting as sleeping neural stem cells. Under the right stimuli, they wake up, divide, and create neuroblasts. These baby neurons then migrate and integrate into your existing brain circuitry. The second niche is the subventricular zone, or SVZ, which is the largest niche. In rodents, cells from here travel to the olfactory bulb to help with smell. But in humans, evidence suggests these cells contribute to the striatum, potentially impacting our motor control and cognitive flexibility. Crucially, these stem cells do not float in a void. They are embedded in a specialized microenvironment composed of blood vessels and support cells. This niche acts as an antenna, receiving signals from your blood. This brings us to the blood-brain connection. Historically, the blood-brain barrier was viewed as an impenetrable wall. But in the context of neurogenesis, it is better understood as a signaling interface. Neural stem cells actually cluster around capillaries to form a vascular niche, allowing them to taste the blood for instructions. The most striking evidence for this comes from heterochronic parabiosis experiments. This is where scientists surgically connected the circulatory systems of an old mouse and a young mouse. The results were startling. The old mouse exhibited a burst of neurogenesis and improved brain function, while the young mouse suffered cognitive decline. This proved that factors circulating in the blood control neural aging. We've identified some of these factors. One is GDF11, a youth factor that declines with age. Restoring it can rejuvenate brain vasculature. Conversely, a chemokine called CCL11 increases with age and actively inhibits neurogenesis. More recently, scientists identified platelet factor 4, or PF4. This is a master messenger released by platelets. It is elevated by exercise and crosses into the brain to reduce inflammation, effectively releasing the brakes on neurogenesis. Now, let's talk about fuel. The brain is the most energy-demanding organ you have, and building new neurons is expensive. This means your metabolism is a primary regulator of your brain's growth. When you are in a state of fasting or carbohydrate restriction, your liver converts fatty acids into ketone bodies, specifically beta-hydroxybutyrate or BHB. For a long time, we thought BHB was just emergency fuel. We were wrong. BHB is a signaling molecule. It crosses the blood-brain barrier and acts as an epigenetic modifier. It inhibits enzymes called histone deacetylases, which results in increased transcription of brain-derived neurotrophic factor, or BDNF. BDNF is essentially fertilizer for your brain. Evolutionarily, this makes perfect sense. If you are starving, you need heightened memory and spatial skills to find food. So fasting triggers a signal to boost your brain power. On the flip side, we have insulin and IGF-1. These are growth signals. IGF-1 is essential for the proliferation phase of new neurons. However, in conditions like type 2 diabetes, the brain can become insulin resistant. This disrupts the signaling pathways in stem cells, leading to reduced survival. This provides the molecular link between obesity, diabetes, and an increased risk of Alzheimer's disease. Perhaps the most empowering part of this science is the muscle-brain crosstalk. Physical exercise is arguably the most robust way to grow new neurons. But how do your leg muscles talk to your hippocampus? They use myokines, which are hormones released by contracting muscle. One of the stars here is irisin. When you do endurance exercise, your muscles produce a protein that gets cleaved into irisin. Irisin travels to the brain and forces the production of that brain fertilizer, BDNF. Mice that cannot produce irisin do not get the cognitive benefits of exercise. It is necessary. Another factor is cathepsin B, a lysosomal enzyme secreted by muscles that correlates directly with improved memory performance. Even lactate, once demonized as a waste product causing fatigue, is actually a preferred fuel for neurons and stimulates the growth of new blood vessels in the brain to support those new cells. Moving down the body, we reach the gut-brain axis. You host trillions of bacteria that function as an endocrine organ. When these bacteria ferment dietary fiber, they produce short-chain fatty acids like butyrate. Butyrate, similar to ketones, can cross into the brain to promote gene expression favorable to new neurons. It also calms down inflammation. Then there is the vagus nerve, the physical highway between your gut and brain. Signals from the gut can trigger the release of acetylcholine in the brain, which calms down microglia, the brain's immune cells, creating a safe environment for growth. Even your stomach gets involved. When your stomach is empty, it produces ghrelin, the hunger hormone. But ghrelin does more than make you hungry. It binds to receptors in the hippocampus and directly stimulates the proliferation of neural progenitor cells. Being hungry puts your brain in a procognitive state to help you hunt and gather. We must also look at the skeleton. The bone-brain axis is a real thing. Bones are not just scaffolding, they secrete hormones. The most notable is osteocalcin, produced by bone-forming cells. Osteocalcin crosses the blood-brain barrier and binds to receptors in the hippocampus, enhancing the synthesis of dopamine and serotonin. Injections of osteocalcin can actually reverse age-related memory loss in mice. This implies that maintaining bone health through impact exercise is directly linked to preserving your mind. Of course, we cannot ignore the endocrine symphony of hormones. Chronic stress is the arch-nemesis of neurogenesis. While acute stress is okay, chronic elevation of glucocorticoids like cortisol is devastating. It arrests the cell cycle of stem cells and causes cell death, leading to the hippocampal atrophy we see in depression. Sex hormones also play a huge role. Estrogen is a potent neurogenic factor, promoting survival of new neurons, which explains the brain fog many women feel during menopause when levels drop. Testosterone supports neurogenesis in males. And during pregnancy, the hormone prolactin surges, stimulating neurogenesis in the maternal brain to prepare for the complex demands of parenting. Protecting all of this is the immune system, the guardians of the niche. The brain has its own immune cells called microglia. They act like gardeners. In a healthy state, they secrete growth factors and prune weak synapses to make room for new ones. But if you have chronic inflammation or inflammaging, microglia turn aggressive. They secrete inflammatory cytokines that block stem cell growth and force cells to become scar tissue instead of neurons. Finally, we have to talk about time. Chronobiology. Every cell in your body has a molecular clock, including neural stem cells. Genes like BMAL1 and clock regulate the cell cycle. If you disrupt these rhythms, you exhaust your stem cell pool. The brain synchronizes this via the release of norepinephrine. Sleep is non-negotiable here. Sleep deprivation disrupts these rhythms and prevents the clearance of metabolic waste that inhibits the niche. So what is the conclusion? The regulation of adult neurogenesis is a testament to the biological unity of the organism. The brain does not renew itself in a vacuum. It relies on the bones for osteocalcin, the muscles for irisin, the liver for ketones, the gut for butyrate, and the blood for platelet factors. This systemic perspective offers a hopeful roadmap. It suggests we can target the brain without touching the brain. By exercising, eating fiber-rich diets, fasting occasionally, managing stress, and sleeping well, we harness the body's intrinsic pharmacy. We are, in the truest biological sense, a holobiont, a single integrated network where the health of the part is inextricably tied to the health of the whole. That is the new paradigm of the plastic brain.